Hello everyone and welcome back to AP Bio with AP Playground. Today we're covering topic 4.3, signal transduction. Objectives today are not too long. We're going to be focusing on the response portion of the signal transduction pathway and the different cellular responses that can arise due to signal transduction, as well as how a change in the structure of any signaling molecule along the pathway can affect the activity of the signaling pathway. And the suggested skill is going to be argumentation, 6C, provide reasoning to justify a claim by connecting evidence to biological theories. Let's begin by talking about effects of signal transduction. And in 4.2, we intro to the signal transduction pathway, talking about reception and transduction, and today we're going to start by focusing on response. But first, a quick review. So when a ligand, the signaling molecule, binds to a receptor, on the cell membrane, then this message is going to be carried throughout the cell through the transduction pathway, usually through phosphorylation cascade or second messengers, until it can elicit a cellular response, usually by altering something in the nucleus. So two of the most common cellular responses are going to be gene expression and metabolic response. So gene expression are these pathways that are going to regulate protein synthesis. They're going to determine whether a certain protein will be produced. They turn specific genes on or off. And when there's a change in the environment that changes the signal transduction pathway, this can change then what proteins are produced, and this can change your outward appearance, your phenotype. And there's also going to be a metabolic response. And this is when the response is within the cytoplasm rather than the nucleus, where there's going to be an affection of protein activity. So enzymes are going to have a change, and they're going to either promote enzyme activity or inhibit enzyme activity through the signal transduction pathway. And another very common response is apoptosis, or programmed cell death, programmed cell suicide, which is very systematic, prevents damage to neighboring cells, but it helps get rid of cells that are potentially sick or old, and also for morphogenesis of hands and feet, taking away the webbing between fingers and toes to have movable appendages. Now talking about environmental and genetic influences on signal transduction. The signal transduction is made up of proteins. The receptors are proteins. A lot of the cascade molecules are proteins, such as the phosphorylation cascade kinases. Those are enzymes. Those are proteins. And so they follow the properties of protein structure. And so pH and temperature can affect these proteins. It can denature them. And denaturation is when the quaternary, tertiary, or secondary structure is broken due to the bonds being broken because the pH or temperature is not in the optimal range. So when this environment changes, the temperature, the pH changes, these proteins can be rendered useless, non-functional, and so the cascade pathway won't work. But also there are genetic influences on signal transduction. Mutations in genes can also affect the proteins of the pathway. And a mutation is just a change in the DNA sequence, a random alteration. And it changes the protein synthesis and what proteins are being produced. It can result in no proteins being produced at all that are necessary for the signal transduction pathway, or for a different protein to be produced that is not what is necessary for the signal transduction pathway. So if you look here, we have a normal gene. Of course, that's going to lead to the synthesis of a normal protein, the necessary protein needed. But if it's a mutated gene and we have a substitution in the nucleotide, for instance, over here, then there could be a change in what protein is produced, or this could result in no protein being produced at all. And we'll talk more about mutations and protein synthesis in Unit 6. But for now, let's continue to inhibitors and activators. And so again, signal transduction pathways are made out of enzymes, and this means that they can also be inhibited or activated, either in the active site or allosteric site. So let's say we have an enzyme here. This is our kinase, for instance, in our phosphorylation cascade, and here's our active site right over here. There can be a competitive inhibitor that takes up the active site, preventing the substrate from entering, or an allosteric inhibitor that attaches to the allosteric site, which will then change the shape of the active site of the enzyme, again preventing a substrate from entering the enzyme. But at the same time, activators also exist that can activate the enzymes within the pathway, such as with insecticides, which are chemicals that activate rapid firing of neurons in an insect's brain, eventually causing the insect to die. Now that we've looked at all the components of a signal transduction pathway, let's look at a specific pathway and analyze all of its parts. So we're looking at the epinephrine pathway, otherwise known as adrenaline. So epinephrine is a hormone that stimulates the breakdown of glycogen into glucose, and glycogen is the stored form of glucose, to increase blood glucose, blood sugar, as part of your flight or fight response. And so it's an example of a signal transduction response to the environment. This pathway is being activated because you're sensing that you're in danger, that you need to either fight or flee. So when we look over here, we have our epinephrine. This is our ligand, it's our signaling molecule, and this is going to be an endocrine system activation because we're looking at hormones that are traveling a long way through the bloodstream. And so over here we have our receptor that's going to be a G protein coupled receptor, because you can see that there's phosphorylation of GTP, and that's going to contribute to the activation of ananyl cyclase, which is going to create our cyclic AMP, which is a second messenger, and it's produced from ATP. And because we have the second messenger, we're able to activate all of these kinases in turn. First protein kinase A, then phosphorylase kinase, then glycogen phosphorylase, and eventually glycogen phosphorylase is going to be breaking down glycogen into glucose. And body is now has the energy and it's ready for flight or fight. And so this is an example of enzymes activating other enzymes. And any part of this could be inhibited. For instance, there's a thing called cyclic AMP phosphodiesterase, which prevents cyclic AMP from being produced. And then what's going to happen is this whole pathway cannot go through because we're not going to get this phosphorylation cascade. So within this epinephrine pathway, we can see reception, we can see transduction, we can see response, and also how the environment, how inhibition can affect the pathway overall. So there you have it, signal transduction review. We have effects of signal transduction that include changes in gene expression, changes in metabolism, and apoptosis. Make sure that you can identify all the parts of the signal transduction pathway. That's extremely crucial. And we also discussed mutations, inhibitors, and activators, as well as the environment and how they can affect signal transduction pathways.